Hi, Abby.
Hi, welcome.
Okay, today we will be finishing the part of bioinformatics and biological data. Just real quick what we have left. And afterwards, I will be talking a little bit about medical terminologies, vocabularies, those kind of stuff. But I will be real quick with that also. So I'm hoping today's lessons won't be that long for 2 hours. Okay. Do you have any doubt from the last session? Maybe something about the sequence alignment or blast or something, because we went real quick at the last part of the previous session when we were implementing the sequence alignments with a blast, that is a search tool, not just an alignment tool, but is to quickly search for similar sequences in huge databases. Do you have any doubt about that, or shall I keep on with the next?
No, I think it's fine.
Okay,
Yeah.
okay, great then. So you can see my slides now. Today we will be talking a little bit about multiple sequence alignment. Then I will just introduce real fast the concepts of phylogenetic analysis, protein structure prediction, gene expression analysis, and genomic analysis. But I will go real fast with those four last points. I just want you to have more knowledge about multiple sequence alignment. I remind you that the previous day what we saw was simple sequence alignment, or pairwise sequence alignment, that is also called. That was when we compare two sequences, maybe of DNA or maybe of protein, 2 by 2. Okay, not multiple sequences, but just 2 by 2 sequences, and we looked how similar they were. I remind you that when we saw that two sequences were quite similar, maybe we could say that it was because they were homologous. Okay. A multiple sequence alignment is just an alignment of more than two sequences. We could align several DNA chains or several protein sequences. That I remind you that when we had DNA sequences were chains of nucleotides, but when we had protein sequences chains, they were formed by amino acids. So the multiple sequence alignment assumes that the sequences are homologous. We are going to align sequences that we already know that are homologous because anything else, or because we assume it. And when we say that two sequences are homologous, I remind you that we were saying that they descend from the same common ancestor. So from an original species that was common between those two different sequences. The algorithms that we will use for multiple sequence alignment will try to find those homologous positions or regions that have the same structure or the same function. As you can see here in this slide, some of the things we make multiple sequence alignments for are phylogeny, that it is the study of the relationships, the evolution relationships between different organisms, to see which domains are more conserved, so they have a very, for example, let's say a stable function, or a very important really function in some organism. Also, as we will see, the multiple sequence alignment provide us tools to predict and compare the three-dimensional, also two-dimensional and four-dimensional structures of proteins. And we can see the promoters that are those part of those genes that are in charge of regulating the transcription of a gene, which promoters are quite highly conserved. Yes, I remind you that this is a multiple sequence alignment. Here is a protein sequence alignment. As you can see, the nucleotides in the chains. Here we are aligning 10, I think. Yes, no, more than 10 sequence here, and you can see that the regions that are more conserved are equal in all the sequences. So multiple sequence alignments are computationally much more difficult than when we were aligning simple alignments, I mean, when we were doing pairwise alignments. It would be ideal to use a similar or an analog algorithm as the Smith-Waterman, that if you remember, it was that that algorithm that was going through every combination of the nucleotides or the amino acids in one chain versus the other, and was giving scores of how much costed to go to one to another, and then it followed the path with the lowest values. Okay, if we had an analog of that Smith and Waterman algorithm that was capable of looking for the optimal alignments in those diagonals of the multidimensional matrix, when we gave a scoring schema, that would be awesome. That would be great. But this algorithm, if we had it, would have to create a multidimensional matrix with one dimension for each of the sequence. So if we, for example, want to align, I don't know, 20 different sequences, we would have a matrix of 20 dimensions, okay? Therefore, the memory and the time that would be required to solve the problem, to align all those sequences, would increase geometrically with the length of the sequences and the number of the sequences, okay? There's not an algorithm that is capable of doing that. So every algorithm that is used for multiple sequence alignment has to be turned into an heuristic method that is capable of solving the problem in a much faster way, okay? So the solution that we might be obtaining at last, it can be not the optimal one, but it is because of that, because we are not going to find the optimus in the problem, but some kind of approach of the heuristics methods that are used. So usually the multiple alignment assumes that the sequences that we are aligning are similar in all of the length of the sequences, and that they behave well like global alignment algorithms. So they also assume that there are not many long insertions or deletions in the sequence that we are aligning, and therefore maybe some sequences may provide us good multiple alignments, but others no. A good algorithm for multiple sequence alignment should align the homologous positions, as we already said, those very highly conserved regions between species with the same structure or function. And this algorithm can deal with sequences that are quite different, but as in the pairwise case that we were seeing the last day, when the sequences are very, very different, they might have problems creating a good alignment. So if, for example, we are trying to align two homologous proteins from two species that are phylogenetically very distant, the proteins are very conserved, but the species are quite different evolutively saying, we might align quite easily the most conserved regions, those proteins, for example, or the most conserved domains of the proteins. But we will have problems when we want to align the whole sequence, the most different regions. This is the same as we saw in the simple sequence alignment, but remember that the multiple alignment algorithms are not guaranteeing us that the solution that we are going to obtain is the best one. These algorithms are not designed to align sequences that do not cover the whole region, like the reads, for example, when we are talking about a sequencing project, what we were discussing the previous day, that when we are, for example, sequencing a whole genome, we make cuts in the DNA material to have a lot of shorter regions, and then we align them to look for the global sequence, but not with this type of alignments, okay? Because these alignments assume that we are aligning sequences in all her region, not just by some parts of the chain. This is what I was saying, that multiple sequence alignments are not suitable for assemblies. Assemblies are those reads that are obtained when cutting the whole genome to sequentiate it, okay? So there are two ways or two kinds of algorithms when we are multiple sequence alignment, okay? There are one type of methods that are called the progressive methods, in which we align the most similar sequences and then we add the other little by little. And we produce a good alignment, but it is not necessarily the best. One example that we will be studying today and implementing today is clustal. I don't know exactly the name in English, how do you pronounce it? Clustal? And then another type of methods are the iterative methods that are more quick and precise, and they build an initial alignment that is continually revised to get the best score. Is a little bit more advanced than the progressive methods. And one example of an algorithm that is used for this is Mappel. So as I said, it is quite common that this type of multiple sequence alignments use a progressive alignment strategy. The methods, when we are talking about progressive methods, are fast and allow us to align thousands, maybe, of sequences, although that won't be what we may want. I mean, when we multiple align sequences, I think thousands of sequences are too many. We normally don't want that much of sequences to align. Before starting the alignment, as we did it in the pairwise case, we have first to decide which is the scoring schema that we are going to follow, with scores we will give to the matrices, to the gaps extensions, what we saw the previous day. The aim of the alignment would be to get the multiple sequence alignment with the highest score, the highest one, as the other day. But in this case, we do not have any practical algorithm that is going to guarantee us the optimal solution. We hope that the solution that we are obtaining is close enough if the sequences are according to the assumptions that we have previously made. So the idea that we have when we first build a progressive construction alignment is to build the pairwise alignment of the more closely related sequences. That is going to be easier for us to build. So we first see which sequences are more similar, and we align those. And then we are going to progressively add the other alignments that we have done till we have the whole alignment with all the sequences. As I said, first we need to determine which sequences are the most close one, the pairs that have the higher similarity. And one fast way to determine that is, for example, to align pair by pairs and see in the single sequence alignment, in the pairwise, let's say, which one has the highest score. So the first step in the algorithm is to create all the pairwise alignment and to create a matrix with the score between all the pairs. This matrix will include the similarity relationships between all the sequences. So when we have the two sequences, we align blocks then. Blocks, I mean already aligned sequences. And then a block against a block. What I was saying before also is that what we do to know which sequences we have to align is to build a matrix of the pairwise alignments score. Here you see that if you have, for example, 5 sequences, you build a matrix with the different sequences for the rows and the different sequences for the columns. The diagonal is 0 because we are not comparing the sequence with itself, and then the rest of the numbers would be the scores that we have obtained of a single sequence alignment, the 2 by 2, the pairwise alignment. We do this because it is easier to align first those sequences that are most similar. Okay. Here, the tree that we obtain from that matrix of sequence can serve us as a guide for the alignment order. Once we have this matrix, we can determine the hierarchical relationship, as you can see here, between the sequences, which are the closest pairs, and how those pairs are related and so on, okay? What we create here is just a hierarchical clustering, a tree. I'm sure that you know more or less hierarchical clustering, and it is what we do here. So, merging two sequence by two sequence. What in clustering we say clusters. We go agglomerating clusters, for example. We can create these trees by using different algorithms. One of the most famous is UPF-MIE, and the other one that we are using a lot in phylogenetics is the neighbor joining. As I said, these type of trees are usually known as guide trees because they help us in the multiple alignment. Okay. Here would be the process. First, we align sequence 1 and sequence 2. Then we align sequence 3 and sequence 4. Then we align the first block that we have obtained, and then the second one with the second one that we have obtained. And afterwards, this huge block, we align it with the sequence 5. And we obtain a tree. These methods are especially biased to fail when the sequences are very different or they are phylogenetically distant. So, cluster W is historically the most used of the progressive multiple alignment algorithms. Nowadays, it's not recommended anymore to use this one because we have other methods that are best for this. Okay, but it was very useful in its days, and it can do multiple sequence alignment. Okay, first what it does is this, building all pairwise alignments to search the scores. Then uses an alignment scores to build the matrix. From that matrix, it generates a tree, and then it does the alignment sequentially from the most similar to the most dissimilar, using the tree as the guide. The most advanced algorithms to date are all based on hidden Markov models, and they have improvements in the guide tree construction. So the most important part of these advantages for these algorithms is that they do better the guide trees when constructing it. And they reduce also a lot the time of computation. One of the algorithms that can create alignment with many thousands of sequences is this one that you can see here, that is a cluster omega. Okay, it's a variation of cluster W, but now is cluster omega. But also keep in mind that no multiple sequence alignment algorithm is going to guarantee us that the alignment that we are obtaining is the best. And especially if there are very different sequences, worst, okay? So, yeah, we are going to check for cluster omega, with protein alignment, because the previous day, if I don't remember bad, we did nucleotides. Okay, yes, we did blast with nucleotides. Today we are going to do a cluster with protein sequences. So I ask you to please go to Teams. You have it here. I uploaded the previous day these frataxin FASTA files. These three from here, that I think that you already have it from the previous day, so you won't have to download them again. And go to cluster omega page. Okay, I will put it to Teams so you. Here, you can please go to the page. and you should be here. Okay.
Okay.
So here you can see that we can do multiple alignment multiple sequence alignments, with different types of sequence, protein, DNA, RNA. Today we'll be working with proteins. Most sequence multiple alignment, it is done with proteins, because what we want to see is how well is domain conserved. So I remind you that we had these three, one second, these three FASTA files. That FASTA was just a bioinformatics way to store the sequences. It can be from nucleotides or from amino acids. And we had one FASTA files with the different mammals sequence of that protein, of the frataxin, that was involved in some iron metabolic processes in the cells, and that is highly conserved between the different mammals. We also have the frataxin mammals closest sequences, like the most similar species sequences, not because of the protein itself, but because they are more close in the phylogenetic global tree, let's say. And then the frataxin protein in distant organisms, those that are not so evolution close. So first, I want you to select the mammals FASTA, okay? In here in the cluster omega, seleccionar archivo, I guess you have choose file if you have in English. Then in the step 2, we can set different parameters. We are going to just let it in the default parameter. Is the output format that we will obtain after the multiple sequence alignment. For example, Nexus give us a tree format with how do you say English, parentheses? I don't know if that's the word in English, but like brackets or so, okay? Is to store the tree that we are obtaining from the sequence alignment, to store it in a text file. Well, different ways of outputting the alignment, the multiple alignment. And there are also more options that we are going to leave as they are. And then we submit our job. It will wait a minute or something. Okay, we have it now. You can see the different organisms that we have inputted here, the mouse, the rat, the dog, okay. And we see them here and the different sequences. Here is what is the alignment itself, but we can also have a summary of the results in all the things that we are going to have from the alignment. The kite tree that we have built, that would be here, okay. We can see the cladogram or the real with the significant distances. Here is the guide tree, as I told you, with the parentheses, or I don't know how that is how is this called in English? Parentheses? I don't know. Also, we can see the phylogenetic tree that is obtained that is not the same because it is built with other type of distance between the different organisms. We can visualize the tree results in different already softwares. And what is you also can here you can copy this direction and you can see the results colored. Okay, no, it wasn't here. Here you have the what it was because I saw it the last day. Let's check 1 second. Here. So colors. Yes, it was this. Okay, in so colors you can see it with the. But it wasn't this what I was going to show you. Because here the only thing that is doing is coloring the different amino acids. But it was no, this one. Okay, it doesn't matter. It doesn't matter. But what I want you to do is to now check yourself for the closest mammals and the distant, also the multiple alignment with the distant organisms, with both of them, okay? And think a little bit about what we obtain and why. I give you 5 minutes. Okay, sorry, I interrupt you 1 moment. I found it where you can visualize the results as I was saying in MView. You click here. Okay, sorry, I was in results viewers from the previous alignment. Okay, and I just clicked view in MView. Here is store our previous alignment, so we don't have to resubmit it again. And we tell it to submit, and we will have it colored. It's just, I think, to visualize it better, but it's the same alignment, okay? So here we visualize which are the most conserved regions. Okay, for example, this one is very conserved. In here we have in the platypus one almost only chain that is only for the platypus. Okay, the platypus will have some kind of features in the proteins that the other organisms do not have. They have maybe some kind of loops or something, but the most conserved regions would be this. Okay, this one here, for example. I don't know, this one here, this one here, okay, all of this. So I'm going to submit also the previous one that I told you, the closest and the distant mammals, so you can see the difference. Well, the distant are not mammals, are a lot of other organisms. Okay, so I'm going to input the closest first. Okay, in the closest mammals, you see that the protein is more similar than before. Okay, for example, the platypus is not here, so we don't have that long sequence without any alignment, for example, or as you can see, more conserved regions between the species are obtained. And we're going to see, I'm not going to output that so we can compare it. We're going to see for the distant mammals, no, distant organisms, sorry, and check whether we have a lot of difference with the closest mammals.
It's not working because you are in MV directly.
Sorry?
It's not working because you are in MView directly. I think you need to go back to the first one.
Here, I cannot put it at the same time, or I didn't understand, sorry.
You need to go to the first tool. No, MView.
Oh, I'm in view. Okay, yes. Sorry, thanks. I have to go to cluster. Yes, that's it. Sorry. Thanks. Okay, you can see here that the alignment is longer because we have a lot of regions that are not matching, so we will insert more deletions or insertions or any what, okay? I just want you to check one thing. Okay, I want you to tell me which species in this protein are closest to human.
Maybe B?
What, sorry?
The B?
The bee?
Yes, no?
Wait, let me 1 sec. But okay, you are seeing for the distant the distant tree, right? You are watching this.
Okay.
Okay, in this, no, right? You were looking in this one. I was looking in the previous one, in the closest mammals alignment.
Okay.
You have it? The human is the most close with the chimpanzee, okay? And also with the rhesus and the macaque that are two other monkeys, also is quite near. Okay, well, just
Yes.
for you to know how to make this kind of phylogenetic tree. Because there are some specialized softwares that are only studying phylogenetics, and you can make multiple alignment with those. But I don't think is that you are going to need that with working with cluster O or cluster W, whatever you can go. So let's continue with this 1 sec. Okay, so phylogenetic analysis, now that you have seen it, tries to infer the evolutive relationships between species. That is what we call to phylogenetic analysis. They try to build the topology that orders the split of the ancestral species that were creating the species that we have today, and the genetic distance that are related to the time that has passed since those splits. Here you can see an example with coronavirus, for example, first with SARS-CoV, that was the first one that we studied, like before all this pandemic and blah, blah, blah. And then with the relationships with SARS-CoV-2, that is the one that we have now, and the different sequences in different places. The phylogenetic methods, usually what they do is to assume that the species that are being analyzed were created by these splits that we are talking of the ancestral species by bifurcating. So if this assumption is not made, the results that we can have in this kind of twists can be misleading. Okay. For instance, if we are analyzing populations within a species, there could be gene flow, so we have a transfer of one gene to one place to another due to the migration, and there won't be reflected in the tree built by the phylogenetic analysis. So, as in multiple multiple sequence alignment, we have some assumptions that cannot be met and that we don't know, and we are interpreting things in the way that are not, but it's something that we have to deal with. So for example, this would be the mammalian phylogenetic tree of the different animals. There were some concepts that I wanted just to comment. For example, the speciation is the evolutionary process by which reproducibly isolated populations can evolve to become different species. Two populations from the same species that are reproductively isolated, that do not have gene flow between them, as we were saying before, can end up with the time creating two new species capable of having sexual reproductions between them, just between one of the isolated population and the other isolated population, but not between each other. Another concept is genetic drift, that is the change in the allelic frequencies. Allelic is just the options in the genes to have one phenotype or another, okay? And is the change of that allelic frequency due to the random sampling of those alleles to create a new generation. There wouldn't be any genetic drift in an infinite population because this random sampling would be extended, okay? So another concept would be selection. Selection is due to the differential reproductive success of different genotypes. Selection removes variation that is created when something mutates, okay? It could be compared with someone that is removing some fragments in a stone when it's making, for example, an statue, to have a metaphor of what selection is. So you can build a phylogenetic tree even when these assumptions that we were talking are not met. That this assumption can be that all of the samples exist, that clades, well, we haven't talked about clades. Clades are not exactly as the same thing as taxons, but more or less is like the branches in the trees. So, that one a branch is a branch, it never merges again with another branch, okay? This assumes that there is not a gene flow horizontally between two species, no recombination, what is really not true. This does happen, okay? Also, interspecific variation is relevant, that different individuals would give the same result even though they have not the same genotype, okay? So just you keep in mind that taxonomy and phylogeny build trees, but they are not the same thing. For example, taxonomy is more in charge of classification, while phylogeny studies the history of the difference in the species, for example. And yeah, that would be more or less all. That trees are used for both, for taxonomy and for classification. For example, taxonomy in the case of diseases is important because we want to have that hierarchical structure of classes, system classification that provide us different hierarchy levels of the diseases. Here what you have represented is a dendrogram. Dendrogram is just a diagram that represents a tree, and we use that term, dendrogram, for general names of trees. And one of the most scopes in which we use that term is for hierarchical clustering. Instead, a phylogenetic tree is a representation of the phylogenetical relationships, just that distinction between a dendrogram and a phylogenetical tree. Also, we have two types of phylogenetic trees that are phylograms and cladograms. In phylograms, the branch lengths represent the genetical distance or the time that it has taken for two species to evolve, and in a cladogram, the branch lengths do not represent anything. Okay, we are just interested in how close are two species, but not how much close, but to which other species are more close than quantifying the closeness of two species. So phylogenetical evidence can be based on the similarity of sequences. We use that similarity to infer phylogeny. So when we have, for example, two analogous genes, can be something misleading for us because two analogous sequences will be developing the same kind of organ, but will be very different in the species or in the evolution process, let's say. So similarity in sequences is usually due to homology, but not always. And it is almost impossible to have a long stretch of similarity due to those analogy in sequences. Phylogenetical trees are usually built from multiple sequence alignments, as we have seen today, and because of that, we assume that the aligned position for its sequences corresponds to homologous positions, and the difference are due to mutations that occur along the evolution of the history. So that's not always true, but is what we use phylogenetic. Why we use multiple sequence alignment in phylogenetics. That's for phylogenetic analysis. I hope that you more or less have the clues of what we do with this and why we do this. In the next point, protein structure prediction is another very huge part of bioinformatics that tries to, from the amino acid chains sequences, infer which is the structure that the protein will adopt. Okay, so from a sequence of amino acids, we want to know in the three-dimensional space which is the structure that the protein is adopting. There are different levels of protein structure. We first have the primary protein structure, that is that chain of amino acids. The secondary protein structure with the alpha helix or the beta sheet. Also the tertiary protein structure where the protein folds into different structures depending on how the side chains of the protein interacts between each other. And then a quaternary protein structure where we can merge different polypeptides or whatever. So what we aim here is from the chain here, know how it's going to be the secondary structure, the tertiary structure, and the quaternary structure. What is not quite easy, but there are several ways to do it. This is an schema of the different things that we have in mind when we do this. But as you can see, for example, one of the first approaches is to sequence align, to multiple sequence align the original protein sequence, because with that sequence we compare in different databases with other proteins that we already know which is their tertiary or their quaternary or their secondary structure, and for that what we do is multiple sequence alignment. So multiple sequence alignment is and also simple sequence alignment is like one very initial step of a lot of bioinformatical processes. Another field in bioinformatics is gene expression analysis. What we do in gene expression analysis is to measure how many copies of a gene we have in different conditions. So maybe the organism is being exposed to I don't know, different conditions. Maybe the organism has the disease, maybe it has not. Maybe we are putting the organism in some temperature on another, or we are exposing it to ultraviolet light or whatever, to see how the levels of expression of that gene are high or low, and therefore we can have more insights of the function of the gene or how it's related to a disease, for example. Or also we can search in which tissues is more expressed or less expressed. In medicine or in human, what we tend to use gene expression analysis for is to check different populations and check in different parts of the body. For example, a gene that is being highly expressed in the I will say in the lung, but in another part of the body it is being low expressed when the organism, the person is meeting a disease or a condition. Okay, so we have an idea of how the gene is overexpressed or low expressed with some conditions or another. This would be an array of different genes and different conditions, and we see how the different genes are, not different genes, but different transcript of a gene, okay, and how they are related between each other. And finally, genomic analysis, what gives us is the annotation of a complete genome, not just a gene or so. And we have here, for example, a possible annotation of the copy of the number variation in humans. More or less, this is all about biological data and bioinformatics. I hope you have like a global vision of what we try to do in bioinformatics and how the data in this kind of approaches is growing and growing, and how we have to know some stuff about biology to be able to interpret it. Okay, so now that we are done with this, I am going to open the other wait here. I will go very quick with this because I think you already have some knowledge about this, if I am right. Can you tell me if you know something about vocabularies and mappings in medicine and in diseases? I mean, do you know any?
Yes, in the other lecture we had a. Sorry, I could hear echo.
Sorry, I didn't.
No. Okay. In the other lecture we did learn about SNOMED, where we talked about this.
Alejandro told you about this moment? Or I did tell you about this moment?
IB2 model. Yeah, we talked about IB2 model and SNOMED, so we have just an introduction knowledge about it.
Okay, great. So I will go very, very fast because I don't want you to duplicate things. Okay? By the way, do you have any question about what we saw in biological data and bioinformatics before I continue?
No, it was pretty clear.
Okay, great.
It's related to other class with using that type of process, but in other fields, so it's great.
Okay, great. So awesome. I really like it, but I think that some of the things are not still very standardized. So people researching on it do what they can sometimes, let's say, because as we don't have still in some parts of it, because some parts are like, I mean, FASTA files is something very common, but it's like an emerging field, and the volumes of data are also very huge. Researchers have to manage with it as they can. Okay, so yeah, I will go with vocabularies and mappings. One sec. Here. On medical terminologies, classification systems, all of these available vocabularies, these control vocabularies that are used in healthcare or in medicine to different issues. We have a lot of vocabularies and a lot of terminologies for different things. We can have it to report some conditions, to more administrative issues, some management things also, to classify and explain diseases, to define treatments, for example. So we can have some protocols when giving medication to patients. All of these are vocabularies and mapping. And I have just summarized it very, because I put it 5 common medical terminologies, but I guess that you know that there are thousands, not thousands, but depending on the country, on the purpose that you want, there are many of them. Okay, so I put it here some definitions, some concepts, but I'm not going to go through them all because I think you know it. Just we have words and terms that a term can be one or more words that are used as a unit. So a term can be several words that mean something. Okay. Also a concept that is used more like an idea or an action or a thing. A synonym that is when two terms mean the same concept. When we talk about terminology, for example, that is a collective set of terms that are used to describe some kind of code set or classification and vocabulary, for example. A code would be a representation of a term, but in a more readily way for a computer or whatever. But in general, most terminologies incorporate a coding system, so computers can process easily, and we have a unique way to describe a term or a concept. Also, a classification that is an arrange or an organization for some kinds of terms. A vocabulary would be defined more like a set of some terms that are more specialized in one field and that can facilitate the communication between different parts of that field. So we are always referring to the same thing, even though we are different parts speaking, let's say. When talking about a vocabulary, there are also different types of vocabularies. We can have a controlled vocabulary that indicates only the set of an individual terms in the vocabulary. We can have an structured vocabulary, or for example a reference terminology that relates to the terms to one another with a set of relationships between the terms and qualifies them with a set of attributes that are associated to the terms, okay? To promote this way the accuracy and the interpretation of that vocabulary. All of this we can model it of course, in different ways. We have different, let's say, manners of representing the relationships and the attributes of a vocabulary, as we were saying. Vocabularies also are said to be mapped to standard terminologies. So a standard terminology is a concept that is more bigger, that is bigger than a vocabulary. Okay. Nomenclature stands for the same thing more or less that vocabulary. Also indexing that would help us finding in those terminologies and vocabularies what we want. Cataloging would be putting away or removing some things that we have. And for knowledge representation, we understand the action of representing the facts in reality. Okay. So how do we represent medical knowledge? This is a really complex problem because several things. One of them is that we have a lot of kinds of concepts to represent when we refer to medical knowledge. Okay, those concepts can be simple or can be complex and can be related among them. Also, the concepts can have units. I mean, for example, for laboratory processes, we will obtain something with units. The concepts can also be negated. Some condition may not have this associated symptom or may not have present that mutation in that gene or whatever. And in a lot of things, time is also involved in the medical knowledge representation. One of the representations that is optimal to medical knowledge is the one of graph, for example, on networks, as we already saw in the diseases networks class. It is necessary to decompose the terms that are used to try to reach the maximum atomacy, okay? As more atomacy we reach, more complexity we can have. And the model we will reach, with the model we will reach, let's say, more comprehensive definitions of the concepts. Also, we have semantic technologies. I don't know if you're familiar with semantic technologies, the semantic web, those ontology stuff that try to model everything in triples, like a graph, like the subject, the link would be the predicate, and the other node would be the object. So we always have the knowledge represented as three relationships. Not three relationships, but a relationship with three, let's say, atoms, okay? Yeah, so yes, these semantic technologies offer a good knowledge representation framework to create relationships between medical concepts, and different ontologies can be created to represent different kinds of concepts in the medical domain. For example, we have the gene ontology to represent information about genes, the disease ontology to represent information about diseases, the I don't know, another ontology to represent laboratory procedures, the symptom ontology to represent the information related to symptoms. So different ontologies that are more focused in the concept or terms that we are modeling. Also, there are several vocabularies and ontologies that are most used than the others when representing biomedical knowledge because they are more standardized, more comprehensive, used in more hospitals, let's say. Okay, one of the most important and largest vocabularies is the one that you told me, Genomic CT, that CT stands for clinical terms. Genomic CT is a collection of medical terms that provide us codes, synonyms, a hierarchical classification, definitions for medical knowledge. Okay, it is used most of all in clinical documentation and reporting, for example, for clinical notes or those kinds of hospital staff. It's considered to be the most comprehensive terminology in the world because it has different languages representation. So if we can, for example, in Spain, the doctors write the clinical notes in Spanish, as they do, we can represent the knowledge in those notes by the concepts in Genomic CT. Here you can search and see in the links that I show you here for different diseases or concepts related to biomedical knowledge in Genomic. Another one is MES. MES is a control vocabulary that is mostly used for indexing journal articles or books in life science. So, for example, PubMed, Medline, also some clinical trial databases. The way that are indexed is by MES terminology. MES, I don't know if I say it, but it's medical subject headings. So it's like, for example, the title of an article, of a journal, of a paper, would be identified by a MES identifier. You have different type of MES terms. As you can see here in the slide, we have, for example, starting for the A would be anatomy. I'm mostly interested in starting with the C for diseases, but there are much of them. And cool things about MES also is that it has different categories. When we talk about diseases for the C, C0.1, for example, is for disease of type whatever. C0.2 is for diseases, let's say, infectious. Other for anatomical diseases. So that's a cool thing of MES, that it allows us to categorize diseases. We have it also by type, that can be infections, new plants, animal diseases, and by systems, respiratory, digestive, cardiovascular. So that's a great thing about MES. Another one would be ICD, that ICD stands for International Classification of Diseases. It's the disease classification that published the World Health Organization, and it has a lot of versions. Now the last one is ICD-11, that you can browse it in the link here. And the cool thing also about ICD classification is that it provides a three-structure identifier. So a part of the identifier will say which category are we talking, another number will say which etiology, another one the localization. So with the entire code, we can have different information about the disease. Also, we have an above category that are the first letter and two numbers of the code that will provide us the chapter and the main diagnosis of the disease that is being identified with that identifier. You can see here all of the diseases the types of diseases that are categorized At first, ICD was primarily thought to be used for the health industry, like for statistical stuff or management issues, but it has been proved that also for scientific researches it can be useful. Another one would be the disease ontology that has been developed as an standardized ontology for the human disease. When all of these terminologies and vocabularies are more centered in human, although they can have some kind of interactions with pathogens or whatever. The concept of disease is modeled around humans. This ontology has the purpose of providing the biomedical community with descriptions for the human disease terms, also the phenotypes, all the medical vocabulary related to disease, as well as the other terminologies were more exhaustive, let's say bigger, more comprehensive. This one is most centered in diseases and also provides a cross-reference to other vocabularies and terminologies, as you can see here. That, for example, we have the corresponding ICD identifier, the MES identifier, NCY identifier. I mean, a lot of them. Okay, so that's great. And one of the biggest is UMLS, that stands for Unified Medical Language System. You can browse some of the things that UMLS provides in the link that you can see here. It will lead you to a full list of sources that are used by UMLS. Okay. It is a compendium of several control vocabularies. Okay, so it is a global system with different parts. MES and SNOMED and GEO, all of that are part of MES. This is like a bigger. The concepts in UMLS are represented by a code that maybe it sounds you, that is called the CUI, the CUI, the concept unique identifier. And also it has different tools, for example, for natural language processing. It can be MetaMap or SEMrupt that allow us text mining, biomedical text. Also, there are several codes regarding lexical terms or string or atomic entities. I mean, they have a lot of things. So, yeah, that will be all. I don't think I am missing anything. No, that's all, guys. I think that's fine. So I'm going to stop sharing my screen. 1 sec. Okay, so yeah. Any questions, comments, suggestions for next year, if you are more interested in anything else that I haven't regarded for these sessions? Or
For me, it was nice. I had no knowledge in genetics, so it's a good introduction to that. And it's pretty related to my other class in data analysis. So, yeah, is fine for me.
Okay, great. Then
Thank you.
has been useful for you. That's awesome.
Yeah, I think also it was a very good introduction. Yeah, as Robin, I wasn't very related to the topic, but I think that yeah, now this is a good way to start looking more after the basics, and now looking forward to the project. I think that will help me consolidate what
Yeah,
I've learned.
yeah, perfect. It will be quite easy. Some simple sequence alignment and some multiple alignment with quite easy questions, and I think that will be all. So don't worry. Okay, so guys, if you don't have anything else, I think for today we are done because it's fine. So nice to meet you, and if you have anything to say, you have my email, my team account, so whatever. Okay.
Yeah, thank you for your
Thank
time.
you.
This was great.
Bye, guys.
Thank you. Bye.
Thank you. Bye.
Bye, bye.